So now my joke is almost ruined, but I was asked yesterday or 2 days ago by Fridtjof whether I was a medical doctor, and I'm not, and that was very disappointing, but I'm still going to use some anatomical knowledge to explain where it is I'm from, because there's some Americans here and they think, Amsterdam, is that in Denmark, or is Denmark in Amsterdam, or why don't we speak Dutch in Denmark, and all that kind of stuff. So just for those, here's Europe, this is the heart of Europe, this is the shoulder. People over in England feel it's kind of cold now, and this is the brain of Europe, where I'm from. So this is what I do. I go out, I find antibodies. This is exactly how it looks. So it gives me a special set. I have a special set of abilities that allows me to look at a picture like this, where people who are not trained, they're like, where's the antibody? It's right there. So many of you in the audience will know this by heart. I have run a research group that has, until last week, been known as the Tropical Pharmacology Lab. We were upgraded to the Center for Antibody Technologies. Here's my people. Very briefly, we work on infectious diseases, special binding antibodies, different formats, all sorts of stuff. But snakebite antivenom is the topic of today, and is probably what we are also most known for. I'm going to take you back 11 years to Tanzania, 2011, and also explain to you a little bit why does a Viking up in the north, who doesn't come across venomous snakes that often, why is he working on antivenom? So I was here basically on safari with my family, but on this trip we came across 2 snakebite victims. A girl, the age of 8, who'd been amputated by the elbow, and a little boy by the age of 5 who'd been amputated by the knee. So obviously this left a very strong impression on me, because we met these at a clinic. And I mean, of course, I wouldn't say of course, but I knew that it was a very bad idea to get bitten by a venomous snake. But I did think that if you reach the clinic, you would be able to get better treatment than amputation, basically. So there's going to be some nasty pictures, just so you're warned. This is not the exact child, but this is a very similar case of how it looks when a spitting cobra that can both spray venom into your eyes and blind you, but which can certainly also bite you and cause severe tissue damage, dermal necrosis. This is how that looks. It was also spitting cobras that had bitten the 2 other children. So classical hallmark of some snakes, that they cause tissue damage. Others, they cause hemotoxicity, or hemorrhage, or inability for the blood to coagulate. Others cause flaccid paralysis caused by neurotoxins. And you could say and if we zoom out and don't just look at the individual victim, we all heard today that snakebite envenoming is one of, arguably, the most neglected of the neglected tropical diseases. It's a major, major problem we typically don't hear much about up here in the north. Going a bit into how do these toxins work, I think Dr. Habib and Dr. Casewell, they showed very well the clinical manifestations and also the venom variation. But if we zoom in on individual toxins, just to get an idea what is the biochemistry, some of the classical toxins to look at are alpha neurotoxins. They target the nicotinic acetylcholine receptor and interfere with, you could say, neuromuscular transmission. Ultimately, that leads to paralysis, including the muscles around your respiratory system, which will cause suffocation and death at the end. Other toxins, we heard John talk about dendrotoxins. They have a little bit the opposite effect. So they work on potassium channels and cause fasciculations, or you could say involuntary muscle contractions. And it might seem counterintuitive that it ends up working the same way kind of as the alpha neurotoxins, because they exhaust all your acetylcholine. So basically you run out of an ability to move around and move your muscles. There's plenty of other toxins. The last example is just a myotoxin, which is a phospholipase that has a completely different function. It goes in and targets cellular membranes, disrupts them, muscles leak out proteins and other things, and these proteins then circulate, end up in the kidneys and cause kidney failure, which is also, I think, one of the classical ways that victims unfortunately die of snakebite. One thing that I think all of you that are not working with antivenom on a daily basis should take away is that there's a difference, at least in the English language, between venom and poison. Poisons are toxic either when they're injected or when they're ingested. And that's because they typically consist of small molecules that can be absorbed in the gut. Whereas venoms are protein-based, they are larger typically than poisons. They need to be injected either by fangs or sort of a stinger. And that means that they are not toxic when ingested, but they certainly are very potent and toxic when injected. Being toxins, proteins, this has, you could say, the implication that we can actually use them for immunization. And we've heard earlier today that existing antivenoms that are on the market are still produced using the traditional method of immunizing larger animals, typically horses or sheep, collecting their blood, isolating the immunoglobulins. And the good thing about it is that it works. It can neutralize venom, but we also heard earlier today that there's a lot of issues when you take something that is, in my view, close to a blood transfusion from a horse. You wouldn't even get a blood transfusion from any random person. Getting a blood transfusion from a horse and injected it into a human being, that has some issues. So you could say what my lab has worked on for the past sort of 6 years, together with John and Anish and collaborators in Costa Rica, is to sort of move into what we call the modern era of biopharmaceuticals and see if we can make antivenoms recombinant. Essentially doing the same as was done with insulin in the 90s or with blood factors, now just with antivenom antibodies. So we work on identifying the specific antibodies, developing mammalian expression systems that can be used in an oligoclonal fashion, so you have one batch to make a defined mixture of human antibodies that can neutralize toxins. Nick showed this figure, and this is just mainly to show that there's a number of benefits if you can move away from antivenoms, but also to highlight that it might be that the future of antivenom might not only be human antibodies, could be that they are a component of future antivenoms, but I also see things like nanobodies and some small molecules as relevant modalities that could be included for broad protection. And I'm not going to go too much into the details, but I think many of you here can imagine there's plenty of benefits moving into recombinant technologies. We use phage display technology that John is one of the inventors of. I'm not going to go into how it works, but just highlighting this is the key technique that we use in my lab. And then I'm going to just also skip a lot of the work we've done in a lot of other animals and take you through essentially just 2 examples to kind of highlight what is the pipeline that we are using. But I'd also like to say we've done not as much as the examples that I'm going to show you, but we've done quite a lot with both snakes, spiders, scorpions, killer bees with Manuela, who presented earlier, and a number of different snakes. I'm going to go very briefly through this the first case because John presented some of it, but I'm going to highlight a few other things that I think are worth realizing when you think of a snake bite. And if you take the black mamba, which is Africa's largest venomous snake, it can reach 4.5 meters in length. It can raise half of its body off the ground. That basically means that it can stand up and look down on a small Viking like myself. It's known for always biting multiple times, always injecting venom. It has a very potent acting venom. And I think herpetologists, and maybe Nick would say there's no snake that is aggressive, but I would say that I hear stories that this is then maybe very proactively defensive. You shouldn't stand between its on its path. That's at least what you hear. So we looked at this snake, and John showed this figure. And one thing that I just want to highlight is that when you fractionate a venom, you get a lot of different protein peaks. And the ones that are marked with 3D structures, those are the ones that are important for mammals. So part of our work, when you talk about what toxins, what targets should you focus on, has been to do what we call toxicovenomics, mapping what does a venom consist of, and which of these toxins are relevant for mammals or for the human case. So we identified a number of these toxins as important. In John's lab, we went through a standard sort of phage display campaign and made monoclonal human IgGs. I'm not going to go into the details because John, he showed it. And then you could say what is more interesting about this work is that we demonstrated using this intracerebroventricular model which was convenient because you use less material. And the dendrotoxins work super well in this one. So it's a convenient way of testing against neurotoxins. But it doesn't reflect the real envenoming. But what we could show was that using a mixture of either 4 or 3 IgGs, we could actually neutralize whole venom in this model. So this was nice work, but it presented a number of different limitations. We did have mice that survived, but it was an artificial setup. So if we move away from just the black mamba and look a bit more broadly, we are lucky to have a Wellcome-funded project where we now are targeting the 26 of the medically most relevant snakes in sub-Saharan Africa. We're both using John's SCFE library, and we're using immune libraries from llamas that we have made. And we target these 18 different elapids and 8 different vipers. To give you some idea about how different are these snakes, here's a sort of a phylogenetic tree where you can see all the cobras being in the big sort of group over here. You have your mambas alone, and then you have the different vipers. But essentially what you want is you want to have broadly neutralizing antibodies. It's certain we will still need oligoclonal or maybe even polyclonal mixtures of antibodies, but if you can make each antibody as broadly neutralizing as possible, you can target these snakes with a, you could say, minimum amount of antibodies. That's going to make production more feasible, cheaper, and is, in my view, absolutely necessary to think that way if you want to do anything about this problem. So we took a different snake, and this is not the same example as John presented, but it is the same snake. We took one of the main toxins from the so-called monocled cobra, Nyacothia, and the main toxin called alpha cobra toxin is relatively similar to one of the toxins from the black mamba. When you look at it and align them, you can see from a sequence, and simple sequence analysis, that these so-called 3-finger toxins that have the same target, they have quite a bit of conservation. So we thought that using a relatively simple cross-panning campaign, where in our phage display campaign we alternated the target between rounds, so that in round 1 we took alpha cobra toxin, in round 2 we took the toxin from the mamba, and then in round 3 we either did another one with the mamba toxin, or we went back to alpha cobra toxin to see if we could sort of force our discovery campaign towards cross-reactive binders. So essentially this is how it looked. We also went down in concentration of the toxins to see if we could really push not just for cross-reactivity, but also for affinity. Very, very simple. I'm just going to say that we improved affinity and we improved cross-reactivity. If you look at the yellow graphs, they are the original. That's the original antibody that I made in John's lab, and then the 2 other antibodies that are highlighted in, I think it's blue. I'm colorblind, so I think it's blue. We were able to make antibodies that had much higher affinity and could neutralize these neurotoxins in a patch clamp assay. That's fine. Most excitingly is that we then tried to do both the standard assays with in vivo, which includes pre-incubation. That's also cheating a little bit when we talk about in vivo models where you mix antibody, toxin, leave them for 30 minutes, and then inject them into mice. That's the graph A, B, and C. They will show you that if you do that, give the antibody the best chances against the main snake. The antibody could protect at a very, very low dose. I think it was just 1 antibody to 1 toxin, full neutralization. Some of the other snakes, you didn't have full neutralization, but we could definitely see we had improved cross-reactivity or broadly neutralizing effects. But what we really wanted to see was, could these antibodies also save mice? So we moved down to the so-called rescue assay, which is what every single reviewer always asks us to do, and what is very complicated to do because it's very, very hard to mimic a real snake bite. But we took mice, injected, in this case, whole venom, so not just the individual toxins. We took the entire venom and injected it, I think it was intramuscularly or subcutaneously, waited not for very long because mice have a super high metabolism, so you need to be very fast. and then I think 10 minutes later injected our IgG, and we were able to save approximately half of the mice. We repeated it without any delay, and I think in that case we could actually save all mice, but it's not so common that you have the antivenom immediately after. But this is the first time we're presenting these results. We expect that we'll have a preprint out soon where you can read more about it. But you could say essentially this is our discovery pipeline where we use in vitro techniques to see if we can find broadly neutralizing antibodies, and then our aim is to see if we can formulate them as oligoclonal mixtures covering even more broadly. These snakes, their venoms are heavily dominated by the specific types of toxins that I mentioned before. Other snakes have many other toxins that we need to go for. So you could see that the mice were very happy if they received the IgG. That's what you should take away. So essentially, this leads me a bit more into what is the vision from here, and that is really if you want to target multiple different snake species, it's not so smart if you just start finding an antibody against toxin A, then you move to B, find another toxin, then you end up with probably a mixture of 300 toxins if you want to target all the medically relevant ones in sub-Saharan Africa. So you really do need to push for cross-reactivity, so you can end up with somewhere between 10 and 30 antibodies to neutralize the sort of entire toxin pool. And that we're working heavily on. Then I want to switch gear a little bit and talk about what is it that we're looking more sort of into the future. What are the main complications? Because we haven't found antibodies against everything, but in my opinion, I think we have sort of established how can we find good antibodies. And I think the main challenge, at least seen from an engineering perspective now, is how do we actually manufacture this stuff? And I'm going to start with just a very, very simple and unscientific slide and say we have a challenge when we talk about recombinant antivenoms. And that is it's relatively cheap to go in and just make another antiserum using the horses. But to keep maintaining horses with all that entails is a relatively expensive process, as Dr. Habib also elaborated on, that this is, these products become so expensive that they are unaffordable for many patients. And it's of course extremely hard to estimate what does a future recombinant antivenom cost, but I want to just highlight that the main issue at this point is the R&D cost. It's not necessarily manufacturing. The manufacturing systems are out there that are at least cost comparable to the existing systems. So we tried to look at, me in a postdoc, at what different formats could be used and tried to make some extremely rough estimates of what would it entail if we used an IgG or a nanobody or somewhere in between. What would the cost of manufacture be, using very rough numbers from industry that were the ones we could get hold of. And this is just to give some perspective of some of the things that you, Dr. Habib, elaborated on, that what is important when you design your antivenom is, first of all, you have to have super good antibodies. You cannot afford to use too high doses. That may be kind of self-explanatory. If you need 10 IgGs per toxin, your price goes up 10x. Cross-reactivity doesn't actually mean so much, but it has more to do with whether it's feasible or not to simply control your process. But assuming you had control in principle, cross-reactivity doesn't matter so much, but it does make it easier to control your process. And then I want to highlight that some of the newer formats that are out there, and especially maybe things like single domain antibodies, they are manufacturable at a fraction of the cost. And antivenom and snakebite is really a numbers game. It's not, there's a limit to how effective you can make your antibody. If it binds to a toxin and doesn't let go, it doesn't get much better than that. So you need, in these cases, as I think both John and Nick had some numbers to, at least with the existing products, to inject 15 grams of protein. That's a lot. So you really need to take into consideration, how do you make this as cheap as possible and as broadly covering as possible? And I really think that looking into some of the more smaller, stable formats is worthwhile. So I'm going to highlight some completely different work. And disclaimer, this is part of a company that I've started doing something totally different in gastrointestinal infections. But here I'd like to propose that looking at some of the simpler formats, such as nanobodies, linking them together into slightly bigger formats, but having the avidity effect, might be worth considering. In this particular project, we took nanobodies that target a specific adhesin on E. coli, and were able to manufacture them so cheaply that you could literally feed them to pigs. Like that is an actual business case we're going for. You always, in Denmark, we have a lot of pigs. We always say if something's cheap, you can feed it to pigs. That is our ambition here. And I think we should not just look at all the fancy cancer medicine that is out there. That's fantastic, and that is really what we are piggybacking on. But we should also look at how can we be inspired by industrial biotech and go for ultra low cost manufacturing systems. And I'm very happy to talk more about this later with some of you if you're interested. Other concepts that could be worth considering is using self-assembly domains and more sort of protein engineering techniques to make ultra-high avidity formats. Here we made both, what is it called, the tetramers of nanobodies that self-assemble. They can be produced microbially. We made 16 mers that can also self-assemble and can bind a lot of different toxins. And by doing this, you of course get a lot of binding sites, and you can alternate the pharmacokinetics, which might be beneficial if you want to have a longer lasting antibody that circulates for a number of days until all the venom has left the sort of venom depot and been swapped up. So this is something we are also experimenting with in my lab. And that's, you could say, the main part of my story. If any of you are interested in talking about diagnostics, I can also mention we made prototypes to determine if you're bitten by a snake, which one bit you. Right now, you can only be bitten by one snake and we can tell it, but we're working on expanding that. And then I want to highlight that the work presented here and all of the work that may not have been presented here but helped lead up to this was the hard work of a lot of people. Not just from my own lab, not only from my good mentors John and Anish at Iontas and Jose Maria and Bruno from Instituto Clodomiro Picaro, but also others in the audience, Manu and many other collaborators at companies like Sophion and other universities. It is really not something you can do alone. And with that, I'd also like to say thanks for your attention, and I'm ready for questions.
Thank you. Questions? So I was wondering, the mechanism of inactivation, so we call it neutralization. So do we need to target specific active sites, or is, for example, binding to a toxin and clearing it via an FC or whatever mechanism also a mechanism?
It's a super good question, and a hard one to answer sort of in black and white. I think in many cases, I mean, if you bind an epitope, the receptor binding epitope or part of a toxin for a neurotoxin, I would say almost obviously that makes sense that will probably neutralize it. But is it enough to bind it at another site and completely change its pharmacokinetics? I think the short answer, no one really knows. But I imagine that many of the toxins, simply by sticking a very big antibody to it, will disrupt its ability to interact fast enough with receptors and might be good enough, maybe. So it's probably, to be on the safe side, good to react with the receptor binding site. But you see with antivenoms, some of them target distal sites on metalloproteases that cause them to change conformation, let go of the ion that's in the catalytic site, and that also has a neutralizing effect. So I think it really depends on what type of toxin you're targeting, and there might be multiple mechanisms of action for how you can. I mean, the simplest thing is sticking a small nanobody that doesn't do anything than just cover it up. Whether improved FC function of IgGs can help neutralize toxins faster, I don't know.
Maybe too slow if you need to act within minutes or hours. Got no time.